Keeling's data showed that carbon dioxide was increasing in the atmosphere by 0.5 to 1 parts per million, ppm, each year. Today, it's rising by over 2 ppm annually, from pre-industrial levels of about 280 ppm to currently over 420 ppm. A concentration of 420 ppm is equivalent to 420 molecules of carbon dioxide among a million air molecules, or 0.0420%. Keeling concluded that the year-to-year increase was caused by humans, mainly our burning of fossil fuels, such as coal, oil, and natural gas. Keeling also observed that carbon dioxide levels didn't just change from year to year. More than a simple line sweeping up and up over time, the Keeling curve is sawtooth, with highs in spring and lows in late summer. Keeling hypothesized that the seasonal pattern reflected the net release of carbon dioxide during winter and early spring when plant growth is slow, followed by net uptake in summer when plants are thriving, using carbon dioxide during photosynthesis. Keeling didn't say anything about microbes. He mentioned land plants to explain why carbon dioxide swings up and down during the year at Mauna Loa, but not in Antarctica. Over 6 decades of additional data have confirmed that carbon dioxide changes more with the seasons in the northern hemisphere than in the south, because the north has more land mass and land plants. Among many reasons why Keeling mentioned only land plants is that his short paper appeared about 3 decades before the importance of microbes in the biosphere was fully recognized. Since Keeling's 1960 paper, many publications have documented the participation of microbes in producing and consuming carbon dioxide and other greenhouse gases. But no book, I suggest, has showcased the many connections between microbes and greenhouse gases, giving these unseen organisms a huge role in setting Earth's climate. Some books have explored how uptake and release of carbon dioxide and methane by microbes led to climate change in the geological past, but we also need a book that explores what microbes are doing today if we are to fully understand the climate change that is now threatening the planet. Of course, we are the cause of this change. Yet the complete story of how the burning of fossil fuels and other human activity is transforming the climate is more complicated and fascinating. It involves the unseen agents, the microbes. What is a microbe? Microbes are organisms that are visible only by microscopy. Collectively, they have nothing in common except for their minute size. The Dutch draper, Antony van Leeuwenhoek, first entered this unseen world in the 1670s when he trained his primitive microscope on samples from a nearby pond, rainwater, and scum from his teeth, and found what he called animalcules, although many of Leeuwenhoek's little animals likely had plant-like properties. Modern microscopy has revealed a few defining characteristics of microbes, most notably the presence or absence of a nucleus, the membrane-enclosed organelle that houses a cell's genetic material. Eukaryotes have one, whereas prokaryotes, that is bacteria and archaea, don't. Eukaryotes include the plants and animals we know from the macroscopic world, but they also include several inhabitants of the unseen world. Among the eukaryotic microbes are protists, such as some algae, protozoa, and amoeba, all single cells. Fungi are also eukaryotes. Even though many are microscopic and some, like yeasts, are single-celled, fungi are not usually classified with the protists. Microbiologists have known about bacteria and protists since the 19th century or earlier. These microbes have been extensively studied because of the diseases they cause, such as cholera, the bacterium Vibrio cholerae, tuberculosis, another bacterium, Mycobacterium tuberculosis, and malaria, the protozoan Plasmodium. But microbiologists didn't know until the 1970s about the existence of another life form on Earth, archaea. Archaea were missed in part because they look like bacteria under the microscope, as both are prokaryotes and lack a nucleus. The difference between archaea and bacteria became clear only when Carl Woese and colleagues sequenced a key gene found in both. In their 1977 paper, Woese and colleagues used eubacteria to denote the typical bacteria well known to microbiologists for centuries, and introduced archaebacteria for the other prokaryotes, represented at the time by methane-making microbes, the methanogens. Woese put archae in front of bacteria because he thought methanogens had a metabolism adapted to the environment.